Gabriel. I'm a PhD student from Steve's lab. And before I begin, I just wanted to ask you a question. Have you ever had a really bad day and things just seem to get worse? Maybe you had a really long day in the lab and you just want to go home, but the bus breaks down or your car breaks down or you drop an entire box of tubes. Well, I had a really bad weekend and I was preparing for this talk and I realized that I was first. So I got really stressed and I decided that I was gonna take a break, go and cut my hair, which turned out to be a really bad idea. And as you can tell, so that's why I got even more stressed and I decided to use my cap as, just like a remedy, a temporary solution for it all. But you see, in parallel, our liver actually has a coping mechanism to deal with hepatic stress as well. So what this involves is a hormone known as activin E. So under normal conditions, activin E is secreted by the liver and it goes through the blood to bind to receptors found on our adipose tissue. So this helps to suppress lipolysis, which is the breakdown of fat into fatty acids. Fatty acids can then be released back into our blood, which can then travel to other parts of our body. So this is usually taken up by our liver, and this leads to liver fat accumulation, and eventually this can progress to a wide range of metabolic diseases. So under stressful conditions, our liver actually releases a large amount of activin E. So this goes on to our adipose tissue as well, and this further suppresses lipolysis. What this would do is it reduces the amount of free fatty acids that can enter our bloodstream, and this reduces the amount of fatty acid that is eventually taken up by our liver. So this would help to alleviate hepatic stress. So then we wanted to understand what types of stresses can activate this hepatic adipose tissue crosstalk. So what we did was we tested different stresses in a human liver cell line, and we found that it was able to increase the gene expression of activin E. We followed this up in mice using injections of different chemicals that can activate these stress pathways, and similarly we saw an increase in gene expression. So we wanted to see whether these results were reproducible, not just in our hands. So what we did was we went to search up published data sets, and we found an increase in gene expression in mice models for type 2 diabetes as well as NASH, which are both known to affect liver function. And we also found an increase in keto diet mice, which also show increased hepatic fat accumulation, as well as in liver samples taken from human fatty liver patients. So together you can see that this hepatic adipose tissue crosstalk does appear to be very regulated by different stresses, and at its core is activin E. So just as how my cap is a temporary measure to help to keep everything under control for a while, or maybe when we are stressed we get a glass of wine or beer or whiskey as a mental health break. Similarly, our liver uses activin E as a measure to keep things under control. Instead of letting your liver get more and more stressed, it is a measure to help to give your liver a little bit more time to deal with the existing problem first. Liver dysfunction is at the crossroads of many metabolic dysfunctions like diabetes, heart failure, chronic kidney disease. Many of it, we know someone that has it. And so with our research, we can understand how our liver manages stress and how we can identify new ways to enhance these stress mechanisms so that we can delay the onset of liver-related diseases. Thank you.